For more than 5 centuries, we thought we understood who the Romans were. Marble statues, Latin orations, the toga-clad senators of a distinctly European civilization. It was one of the most settled assumptions in Western history, repeated in textbooks, embedded in museum exhibitions, reinforced every time someone traced a line from Rome to London to Washington. The Roman Empire was the foundation stone of European identity. But in 2019, a team at Stanford University cracked open the skulls of ancient Romans, extracted powder from a bone smaller than a walnut, and fed it into a gene sequencer. What came back shattered the assumption. Because during the very centuries we consider the height of Roman civilization, the people living in Rome, genetically, looked almost nothing like modern Europeans. They looked like the people of Syria, of Lebanon, of Greece and the Eastern Mediterranean. And somehow, for hundreds of years of scholarship, nobody had seen it coming. The old story was elegant. Rome begins as a cluster of hilltop villages along the Tiber around 753 BCE. It grows into a republic, then an empire stretching from Britain to North Africa to Mesopotamia. At its peak, that empire governed somewhere between 50 and 90 million people across 3 continents. The city of Rome itself became the first settlement in the ancient world to surpass 1 million residents, a threshold that no European city would reach again for 1,500 years. And everywhere that empire went, it carried its culture. Latin, law, engineering, the very concept of citizenship. The assumption, reinforced by centuries of classical scholarship, was straightforward. Romans were Italians. They were the genetic and cultural ancestors of modern Europeans. The empire projected its own people outward, and the populations it absorbed were gradually Romanized. Immigration existed, of course. Historians knew about slaves, traders, and foreign legions, but these were seen as footnotes. The core population, the people who built the empire, were understood to be rooted in Western European stock. This felt settled. Archaeological evidence supported it. Roman burial customs, architecture, and inscriptions all pointed to a culture deeply anchored in the Italian peninsula. And because no one had a way to test the genetics directly, the assumption survived unchallenged for generation after generation. But there was a weakness in the story, one that hid in plain sight. The problem was that scholars had always studied Rome through its objects, not its people. They read inscriptions. They cataloged pottery. They mapped trade routes by tracking amphora fragments across the Mediterranean seabed. All of this told a rich story about what Romans made and moved. But it said almost nothing about who they actually were at the molecular level. The bones were there, stored in museum collections and university basements across Italy. Thousands of skeletons from dozens of excavation sites spanning millennia. But for most of the 20th century, extracting usable DNA from ancient remains was considered nearly impossible. The molecules degrade, they fragment. They become contaminated by the DNA of bacteria, fungi, and every archaeologist who ever handled them. For decades, attempting to sequence an ancient genome was like trying to read a book that had been shredded, soaked, and buried for 2,000 years. And so the assumption held. Not because the evidence confirmed it, but because the technology to challenge it simply did not exist. The breakthrough came from an unexpected place, a tiny structure deep inside the human skull called the petrous bone. It sits at the base of the cranium, protecting the cochlea, the spiral organ of the inner ear. It is, by a wide margin, the densest bone in the human body. And that density turned out to matter enormously. Because while every other bone in a buried skeleton slowly surrenders its DNA to the soil, the petrous bone locks its genetic material inside a fortress of mineralized tissue. In some cases, researchers found it preserved up to 100 times more human DNA than teeth, femurs, or any other skeletal element. Ron Pinhasi, an evolutionary anthropologist at the University of Vienna, was one of the researchers who helped pioneer the technique. The process itself is precise and almost surgical. Working in a clean room designed to prevent contamination, a researcher uses a sandblaster to carefully expose the cochlear portion of the petrous bone. The bone is then ground into a fine powder, roughly 5 micrometers in diameter. That powder is dissolved in a chemical buffer, and the ancient DNA, still clinging to its mineralized host after millennia underground, is extracted and prepared for sequencing. Before we see what this technique revealed about Rome, if you are finding this discovery as fascinating as we are, subscribe so you do not miss future stories like this one. It was this method that allowed Jonathan Pritchard, a population geneticist at Stanford, and his collaborators, including Alfredo Coppa at Sapienza University in Rome, to attempt something no one had done before: a full genetic history of the city of Rome. They gathered 127 ancient genomes from 29 archaeological sites in and around the city. The samples spanned 12,000 years, from Mesolithic hunter-gatherers who roamed the Apennine Mountains around 10,000 BCE all the way through medieval times. 33 of those individuals were directly radiocarbon dated. The rest were placed in time by their archaeological context, the layers of soil, the style of burial, the objects interred alongside them. The early results told a familiar story. The oldest genomes, 3 hunter-gatherers from a cave called Grotta Continenza, matched other Western European populations of their era. Then, around 7,000 to 8,000 years ago, a massive wave of farmers descended from early agriculturalists in Anatolia, present-day Turkey, arrived and reshuffled the region's genetics entirely. A second wave followed during the Bronze Age, bringing steppe ancestry from what is now Ukraine. By the traditional founding of Rome in 753 BCE, the population looked roughly like modern Mediterranean peoples. So far, no surprises. Then the team reached the imperial period, 27 BCE to 300 CE, and everything changed. Of 48 individuals sampled from this era, the genetic profiles were overwhelming. Only 2 showed strong ties to Western Europe. 2 more carried North African ancestry. The remaining 44, the vast majority, had genetic signatures linking them to Greece, Syria, Lebanon, and other parts of the Eastern Mediterranean and the Middle East. On a principal component analysis, a kind of genetic map, these imperial Romans clustered not with modern Italians or French or Germans. They clustered with modern Greeks, Cypriots, Syrians, and Maltese. This was not a subtle signal. It was a transformation. Consider what that means. During the very centuries that gave us the Colosseum, the Pantheon, Roman law, and the concept of the Republic, the genetic heart of Rome had shifted dramatically eastward. The people walking the streets of the world's largest city, buying grain at the Forum, attending the games, they carried in their DNA the deep ancestry of the Levant and Anatolia, far more than the ancestry of the Italian peninsula itself. 3 layers of evidence reinforced the finding. First, the sheer scale. This was not 1 or 2 outliers. It was the dominant pattern across nearly every imperial era individual in the dataset. Random chance could not account for 44 out of 48 samples clustering in the same ancestral region. Second, isotope analysis provided a critical check. At the site of Isola Sacra, 9 individuals were tested. 4 fell into a Near Eastern genetic cluster, 4 into an Eastern Mediterranean cluster, 1 into a European cluster. And yet their oxygen isotope ratios, which reflect the water a person drank during childhood, were all compatible with having grown up locally. These were not recent arrivals. They were second generation, third generation, perhaps deeper. The diversity was not transient, it was structural. Third, the pattern ruled out the most obvious alternative explanation, that this was simply Rome importing slaves from the East. While slavery certainly played a role, The isotope data and the breadth of the genetic signal pointed to something larger, sustained voluntary and involuntary migration that reshaped the city's entire demographic foundation over centuries. But why had no one predicted this? The mechanism of error turns out to be revealing. Classical historians had always known that Rome was connected to the East. Greek was the empire's second language. Temples to Syrian and Egyptian gods stood in the city center. The earliest known European synagogue was established at the port town of Ostia, but this evidence was treated as cultural contact, not demographic replacement. The implicit assumption was that a few thousand merchants, priests, and diplomats had brought their customs to Rome, while the underlying population remained essentially Italian. Archaeology reinforced this by focusing on what people made rather than who people were. A Roman-style burial with Latin inscriptions was classified as Roman, regardless of the ancestry of the person inside the tomb. And because the tools to test genetics directly did not exist, the conflation of culture and biology went unchallenged. There was also a structural bias in historical sources. Most of what survives from the Roman period was written by elites, senators, emperors, and poets who represented a tiny fraction of the population. Their world was Latin-speaking, politically Italian, and self-consciously Roman. But the genetic data suggests that this literary class may not have been representative of the city they governed at all. The follow-up studies only deepened the picture. In 2024, a team again led by Pritchard published results in eLife, this time analyzing thousands of genomes, including 204 newly sequenced individuals from 53 archaeological sites across 18 countries. The findings confirmed that diversity was not unique to the capital. At least 8% of all sampled individuals across the entire empire were buried far from their genetic homeland. Sardinia, the Balkans, and parts of Central Europe all showed striking genetic variety. The empire had not just connected cultures, it had physically moved people across continents within their own lifetimes at a scale that would not be seen again for centuries. Meanwhile, a 2023 study led by Inigo Olalde and David Reich, published in Cell, delivered perhaps the most provocative finding of all. Their team analyzed 136 individuals from the Balkans, a region that served as a crucial military frontier for Rome. Despite centuries of Roman military occupation, cultural influence, and administrative control, the DNA showed no genetic contribution from people of Italian descent. None. Instead, the influx came from Western Anatolia, another part of the Roman Empire entirely. Even more strikingly, among the burials at Viminatium, one of Rome's largest frontier cities in present-day Serbia, researchers found a 16-year-old male of 100% East African ancestry. He had been buried with an oil lamp depicting Jupiter's eagle, a symbol of Roman religious life. A teenager from sub-Saharan Africa, living and dying in a Roman city along the Danube, integrated enough to be buried with Roman iconography. Not everyone agrees on what these findings mean. Some scholars caution that the sample sizes, while impressive for ancient DNA studies, still represent a tiny fraction of the millions who lived in Rome. Others point out that the petrous bone technique introduces its own bias, since it requires intact skulls, which are not always available or representative of the broader burial population. There is also debate over how to interpret the Eastern Mediterranean signal. Kyle Harper, a Roman historian at the University of Oklahoma, has noted that the eastern provinces of the empire, cities like Alexandria and Antioch, were simply more densely populated than the western provinces. The genetic shift toward the east may reflect population gravity more than any deliberate policy of migration. The people did not come because they were invited. They came because they were the majority of the empire's urban population, and Rome was the ultimate destination. Others have raised questions about timing. A 2025 study analyzing a Republican-era individual from Tarquinia found that Eastern Mediterranean ancestry may have been arriving in central Italy 200 years earlier than previously thought, during the late Republican period, well before the formal establishment of the empire. If true, this would mean the genetic transformation of Rome was not a consequence of imperial expansion. It may have been a precondition. What remains undisputed is the trajectory. Before Rome's rise, the region's population was genetically European. During the empire's height, it was genetically Eastern Mediterranean. After the empire's collapse, when Rome's population plummeted to roughly 100,000 and the Visigoths sacked the city in 410 CE, the ancestry shifted back toward Western Europe. By medieval times, Roman residents once again genetically resembled the populations around them. The Eastern Mediterranean signal had almost entirely vanished, replaced by Central and Northern European ancestry, carried in by Lombards, Goths, and the political realignment of the Holy Roman Empire. The implications cut deep. If the people who built and sustained the Roman Empire at its zenith were genetically closer to modern Syrians and Lebanese than to modern Italians, then the entire framework through which we have understood the classical world requires recalibration. The story is not wrong, exactly, but it is incomplete in a way that changes its meaning. Rome was never a single people projecting outward. It was a system that drew the world inward, a gravitational center that absorbed, mixed, and remade populations on a scale we are only now beginning to measure. Today, the petrous bones of ancient Romans still sit in clean rooms across Europe, each one a sealed archive waiting to be read. Every skull opened adds another data point to a picture that grows more complex and more human with each genome sequenced. The civilization we thought we knew best turns out to be the one we understood least, not in what it built, but in who built it. If this hidden chapter of history intrigued you, subscribe to explore more mysteries. The evidence is only now revealing. Here is the question that keeps researchers up at night. If Rome, the most documented civilization in ancient history, could conceal the genetic identity of its own people for 2,000 years, what else might we be wrong about?